bit about our speaker today, Dan Rokhsar. Dan is the program head for computational genomics at the Joint Genome Institute, but in addition, he follows in the considerable footsteps of Lawrence in that he is trained as a physicist, an undergraduate degree from Princeton, and then a PhD in theoretical physics at Cornell University before going on into a postdoctoral fellowship at IBM. So it was later that Dan shifted his research interest from materials and physics into biophysics and ultimately segued into computational biology and genomics. And it was back in the year 2000 that he was invited to join the Joint Genome Institute and lead its computational biology effort. And computational biology sounds like so many words jumbled together, but really it's one thing to characterize the genome or the genetic code of an organism. It's another thing to translate that code into the functional units that we can better understand how these natural systems work together. So Dan was a former NSF Presidential Young Investigator, also a recipient of a Sloan Foundation Fellowship, and a Miller Research Professor and a Guggenheim Foundation Fellow. He also serves as the JGI Eukaryote Program Head, and that's another fancy word simply to mean that our larger genomes that we're tackling, not those little squidgy things, but rather plants as eukaryotes, and also fungi are eukaryotes as well, and generally have larger genomes than bacteria. So he's been really driving the plant portfolio, the biomass portfolio of the JGI. So Dan is here today to present his views on the genomic advancements that are going to improve biomass for biofuels. And so if you would please join me in welcoming Dan Rokhsar.
Thanks, David, and thank you all for coming. So I'm gonna stand here because last time I was in this space, I saw Josh Kornbluth speak, and so I'm not going to be as funny as him, but at least I'll stand where he stood. Maybe that's more important than following the footsteps of Lawrence. So I'm going to tell you about some of the work that's going on under the general umbrella of the Helios project relating to bioenergy. I think I just turned something off. And I want to, the takeaway that I want you to have at the end of this hour is some understanding of what is bioenergy from biomass and what are the things we're trying to accomplish. Then what is genomics and how that might play into improving our ability to make biofuels. And then a few vignettes at the end about some of the progress that we've made in the last few years, as David mentioned, looking at different genomes and trying to use that information to help accelerate this process. And there'll be a lot of interesting questions, both technical and sort of socioeconomic questions, and we can talk about those at the end. So Josh didn't have to do this. Oh, there it is. I was going backwards. Okay, so this is what I want to get to, sort of talking a little bit about bioenergy, and I kind of assume that this audience, many of you have been to these Helios lectures before. You'll probably have a good sense from either those lectures or other things you've heard, what is bioenergy and what are the things we're trying to accomplish. But I want to bring everybody to the same basic level. Then also I want to talk a little bit about what genomics is and how genomics came to be involved in this bioenergy endeavor, specifically how it came to be involved here at LBL and through the Helios project. And then, as I said, some vignettes, some early progress. Okay, so just to orient ourselves, obviously we're a very energy-intensive society. And you can see here, this is sort of a failing pointer, but you can see that maybe 150 years ago, the energy was generated from wood. We went through a phase where coal was king, and now it's coming back, unfortunately. And then petroleum use is rising. These are feeding our never-ending need for different kinds of energy, with the main contributors being wood, coal, and now petroleum. So it's kind of different ages of the industrial revolution. And of course, as all of you know, what that does, among other things, is generate a lot of carbon emissions. Those carbon emissions go into the atmosphere and create various kinds of problems. And if you follow the black curve here, that's the total emission from all of these sources, and it basically is continuing to rise. And so one of the things we'd like to do is feed the growing world economy with energy, but somehow avoid the catastrophic effects of this curve continuing to go through the roof. So where does all this is carbon emissions from fossil fuels, where does it go? Well, first, where does it come from? It comes from carbon that was deposited hundreds of millions of years ago by decaying plant material to form coal and oil deposits that today we take out and then burn them, and in the process release that carbon back into the atmosphere. So we're basically pumping carbon from these long-term stores inside the Earth up into the atmosphere. From the atmosphere, it can be then absorbed back by plants, or some amount of it gets absorbed into the oceans. And there's this global balance that can withstand some amount of additional carbon into the atmosphere, but is getting saturated. Okay. So the release of the carbon from these fossil fuels is something that we'd like to defeat. And one way around that is to take advantage of a very natural process, the process of photosynthesis, the process that David referred to earlier. So this is Plant Biology 101. Light energy comes in from the sun. Carbon dioxide is present in the atmosphere. Water is absorbed by the plant. And the combination of those three ingredients gives rise then to a growing plant, which means that carbon dioxide is incorporated or fixed into the material that makes up the plant. In the process, oxygen is liberated. But the main point that I want to make here is that this process of photosynthesis draws carbon dioxide from the atmosphere and embeds that carbon into the plant material. So if we then take that plant and just simply burn it, the combustion process basically liberates carbon dioxide, but it's carbon dioxide that we had already taken from the atmosphere. So it's kind of a net neutral process. And so if we could continue this process, what we're doing is continuing to get energy from the sun, incorporate it into plant material, and then liberate it through some kind of controlled burning process that we as humans can use. Okay, so it's a renewable energy source, and if you could really do it in the simplified way that I've just described, you would have a closed circuit of carbon, and we would not be contributing any more carbon to the atmosphere. Okay. That's a little bit of a naive perspective. Of course, it takes more than just sunlight, carbon dioxide, and water for a plant to grow. And so a lot of what we're going to try to understand is how we can optimize this process. Okay, so this is, in words, what I just on the screen, what I just said out loud. Burning fossil fuel takes carbon that's already been sequestered away and releases it into the atmosphere. The natural process of plant growth, taking carbon dioxide, water, sunlight, and various nutrients like nitrogen, phosphorus, potassium, and so forth, if we burn that, we would be recycling carbon dioxide. Of course, not all the plant material gets burned. Some plant material is deposited underground in the root systems. Leaves fall down, they get degraded by microorganisms and are left in the soil. And so there's also the effect of pumping carbon dioxide from the atmosphere into the soil. Another way and not in this closed cycle, but in a way that is withdrawing the carbon. So there's this idea of CO2 capture in roots and soils and so forth. So there's a technical question, which is, can we take advantage of this very simple biology, simple in principle biology, to harness it for human uses? Can we do this efficiently and minimize other impacts on the environment? And if the answer to that technical question is yes, then we have a bunch of policy questions. If it is feasible, how do we go about doing it? Do we want to do it as a society? What are our other options? And so I'm going to focus mostly on this technical question. How do we figure out whether we can do it efficiently? How can we minimize the impact on the environment? But of course, it all feeds together into this technical and political discussion. Okay, so how do we do this now in a large-scale process that gives us transportation fuel? Well, everybody has heard about ethanol from corn, and the way that works is energy from the sun comes, it's absorbed by the plant. Corn as a food is optimized for storing starch in kernels. And we'll see a little bit about where that came from. So the starch-rich kernels can then be converted into their constituent sugars. Those sugars, like any sugar, can be fermented and produce alcohols. And under the right conditions, you can get ethanol, which then can be mixed with gasoline and fed into a conventional car as long as you don't use too much of it. Okay. So this is a process. The advantage of it is that it's something that already exists. We already know how to work with corn kernels and get them into starch and work with the sugars. The disadvantage is that this is not a very efficient process for several reasons. One is we're only using part of the plant. And another reason is that corn is already a plant that we're growing for other purposes. We're growing it for food purposes. So we'd like to figure out ways that we can use the same basic process. Sunlight and carbon dioxide gets absorbed by plants and then converted into fuel, but do it in a way that is more efficient than just working with the kernels and is also fitting more in with our ideas of what kind of land use we might want to have. Okay, so these are some of the problems. Corn, because it's been optimized to produce these big ears of corn with these large starch-rich kernels, is very water-intensive to grow. It's very fertilizer-intensive. It's an annual crop, so you have to plant it every year, as opposed to a perennial grass, which might just grow year after year without the initial planting. There's the issue of land use and the food versus fuel. And then there are some problems with ethanol. Ethanol is not probably not the fuel of the distant future, in part because it's difficult to pipe it from a production plant. It doesn't have the same kind of fuel value of gasoline. In fermentation, the limits of fermentation are that eventually the microbes, think about the microbes that ferment grape sugars into wine, or sugars from other grasses into beer, those have a limited alcohol content. And part of the process, part of the reason for that, well, we wouldn't be able to tolerate it if we were drinking 200 proof alcohol, so it's good for us. But in the same way, it's also toxic for the microbes. So there's a limit to how good you can do in enriching the fermentation product for ethanol. And so if you want to put that into your car, you have to withdraw all of the water, and that takes energy to distill the water out. Okay, so one of the big energy-intensive and fuel-intensive costs in this process is the significant energy input that's required to purify out the ethanol. So these are all kind of issues that we'd like to avoid and move on to sort of the next generation, probably as soon as we can. So if you think about this process of carbon dioxide being absorbed by the plant with sunlight and water and making plant materials, if you look at a corn plant, yes, it has these big ears of corn, but a lot of the other material of the plant is also produced by this process. So a lot of the carbon that the plant has fixed into its material is not in the corn itself, in the ear of corn, but it's in the rest of the plant. Okay, and this is sort of a view of what different plants look different under the microscope, but this is a section of a pine board after it's been dried, and each of these little cell walls here, if you looked at them more closely, you would see these fibers, and each fiber is a polymer of, in the end, sugars. So the sugars that are made during photosynthesis get incorporated into these kinds of polymers and ultimately give the plant its structural rigidity and the main mass of the plant. So when we talk about biomass, we're basically talking about the totality of the plant material that's been generated by photosynthesis. And the idea would be that these are, in the end, just a fancy way of the plant storing its sugar. We want to get at that sugar in the same way that we get at the sugar that's in corn kernels and then break it down. So it's a very, in a lot of ways, it's a very conceptually simple thing to do. One of the problems is that plants have evolved to resist breakdown by microbes or fungi. Okay? They would like to live a long time, or at least long enough to reproduce. And so this structure is actually very difficult to break down enzymatically, in part because plants have evolved to be that way. So we have to work against this sort of natural challenge. Okay, so the idea of biofuels from biomass is basically to use the whole plant. And if you can use the whole plant, then you're no longer restricted to working with corn. And corn, remember, was optimized because the kernels had been selected to have a lot of starch content. You can work with any rapidly growing plant. So this is a regular-sized person. And this is a field of a particular kind of perennial grass called miscanthus. And that's the growth in 1 year. Okay? So if you could figure out a way, and there's nothing analogous to, there's something analogous to it, but there's nothing that has remotely the starch content of an ear of corn. even though this is closely related to corn. We'll talk a little bit more about that, but the idea is to take these very rapidly growing plants, some of which optimally could be grown on various kinds of land that we wouldn't even think of using for food. They wouldn't be suitable for what we can think of as agriculture. Maybe they could be used as a way of producing enough biomass that then we could break down and get various kinds of fuels. So how do we go about thinking about this problem technically and scientifically? Oops. Okay, so the advantage of this is similar kinds of pictures. The advantage is that there's a lot of this material out there. It's by definition renewable as long as you set up your cultivation practices properly. And because these are the kinds of plants that grow out in the prairie, for example, or under various kinds of conditions where you're not actively trying to carry out agriculture, they don't need as much water, they don't need as much fertilizer. They're growing in very natural mixtures of different kinds of plants, living along with animals and various other kind of commensals. So these are sort of very natural kinds of materials, and they don't need as much water or fertilizer. We have, depending on how you look at it, either optimized corn, or corn has optimized us to take care of it, as Michael Pollan said in his Botany of Desire book. Corn is not something that would survive without all of the inputs that we give it, corn as we know it today. But these are things that grow without a whole lot of attention from us, and we like to figure out, can we use them? Another advantage to thinking about this is that if you broaden your perspective on what biomass is, basically anything that's produced from plant material is ultimately biomass. So any kind of byproducts, waste wood, waste paper, a lot of waste materials ultimately are made out of these materials derived from plants. And we could also conceive of breaking them down and converting them to some kind of fuel. Okay, so what do we need to be able to do this? Well, we need to have a better understanding of how plants make this material called cellulose, or lignocellulose. This is the actual technical name for the plant material. So we'd like to understand how those cell walls are made naturally, and also to understand the whole totality of what it takes to grow a plant, because we want to use that information. And then after we've grown our biomass in a very ideally sustainable way, we want to advance to the next image. And we want to figure out ways then of breaking that down efficiently, ways of taking this polymerized sugar and converting it back down into individual sugar molecules that then can be fermented. Okay? And so there's two different things. We want to first build up the material and then break it down. Both of these have some connection to genomics. Okay, so what is genomics? Well, I think everybody here has some familiarity with the Human Genome Project. It's been in the news over the last decade quite a bit. It's a project that was actually started partly at Lawrence Berkeley Lab and the other DOE laboratories in the mid-'80s and came to fruition in the middle part of this decade. It was a big international effort to determine the sequence of the genes of sort of a typical human being. And now we're in the process, or the larger international community is in the process of figuring out how the individual genomes of everybody in this room differs from each other. What are the patterns? What are the kinds of variations that we see, and how does that relate to individual susceptibilities to disease and other sorts of questions? One thing to take away is that the difference between any individual in this room and the person sitting next to them is about 1 out of 1,000 of the letters of the genome. We often talk about the genome as being a string of individual letters. And I'll show you that on the next slide. So in the process of doing this, this international community built up a capacity for sequencing genomes. So that's determining the base sequence of the DNA in a particular organism. And once you figure out how to do that in the human genome, you can then use the same technology to figure out how to get the genome of pretty much any other organism. And that's one of the things that we do at the Genome Center. I'll show you a picture in a few minutes. Okay, so here's another view of the same kind of thing. So the genome is contained in chromosomes. These are the 23 pairs of chromosomes in the human genome. And if you looked at each chromosome and zoomed down, you would see basically a string of chemical units. And there are 4 kinds of chemical units. And we call them A, C, T, or G for their chemical names. So really, this is sort of Genomics 101. All you really need to know is, if I wanted to know what the genome of an organism looked like, it would typically look like this. This happens to be the insulin gene that I just downloaded off the web. All this information is freely available, which is a nice feature of this international collaboration. And once you have access to this sequence, you can then take advantage of it to do various kinds of experiments, in this case relating to how the insulin molecule is made, how it signals to other cells, how the cells receive that signal, and how the whole system works. So this is very powerful. Even though it kind of looks like gibberish, it's a very powerful piece of information that then accelerates anybody who wants to do work with this molecule. It will take a few lectures to tell you the details about what exactly is a gene and how do you recognize genes in the genome sequence. Suffice it to say that in the human genome, there's about 20 or 25,000 individual genes. And there's a comparable number in plants. Plants may have a little bit more than humans. Okay, so what good is it? Just as a string of letters. Well, it allows you to do things knowing the complete genetic makeup of an organism. So if I want to look at a human cancer, I now have a list of all of the genes that are in the human genome. I can ask in a systematic way, in that cancer, which of these genes is inappropriately turned on or which of these genes is inappropriately turned off. And I don't have to do it in a one-at-a-time way like people were doing it 10, 15 years ago. I can ask that question of the entire genome in one experiment. And the reason I can do that is because people put in the effort to get that infrastructure of figuring out what the genome sequence was. Another thing that's a little bit more subtle than that is you can start to understand how sequence variation correlates with function. So I think everybody's familiar just from opening up the newspaper or listening to NPR, however you get your news on the web. Every couple of days, there's a new gene, discovered for some human feature, some human disease. And in the same way that that's happening in the human biomedical research community, the same kinds of things are happening at a slower rate in the plant science community. So if I look at different plants, they're the same. Maybe I look at the same kind of plant. I look at a bunch of eucalyptus trees around this area. Some of them seem to grow robustly. Other of them have succumbed to disease. Some of them grow rapidly. Some of them grow slowly. Some of them seem to only thrive under conditions where they have a lot of sunlight or a lot of water or a lot of some other kind of nutrient. Others are able to grow under harsher conditions. Some part of that variation is due to differences in the genetics in the genes of the organisms, not so much at the level of how many genes they have, but at the level of subtle variations in this code. Okay, so one of the things we'd like to understand is how do variations in the genetic code give rise to differences between different plants? And we'll see why that's important in a second. Okay, so I talked a little bit about bioenergy and the general idea of using biomass as a source material. I've talked about what a genome is. And now we want to try to synthesize it and figure out what is it that we want to do with biomass crops. We want to grow these plants so that they're well suited to our industrial processing in a way that doesn't damage the environment or is sustainable in its environmental impact. And one of the sort of a lesson that we can get from this is going back to other examples where we've taken wild plants and we, the human race, has taken wild plants and improved them for a particular purpose. And one example, sort of an instructive example, is the example of maize. So maize was domesticated in Central America about maybe 5,000 years ago. And if you go to Central America, I haven't done it, but I've seen pictures, and you look at the ancestral maize plants, they don't have anything that looks at all like the ear of corn that you're used to buying in the store. Okay. They're sort of scraggly plants. They have little tiny collections of seeds that might only have dozens of seeds in them. But through the process of domestication, Native Americans, through relatively few genetic changes, identified natural variants that were out there in the population. And through a process of breeding those natural variants together, we're able to produce plants that had much more robust ears and also a bunch of other features. Instead of, if you look at a modern corn plant, it's basically a single stalk with ears coming off of it. If you look at the native teosinte, they're these kind of scraggly plants with lots of branches. So in the process of domestication, we have basically converted the energies that the plant spent normally on creating this very, very branched structure into something that has much less branching, and in that way diverts less energy from making the corn, which is what we want to be eating. Okay. And this was not a process that involved genetic modification in any sort of evil sense. This is just basically natural breeding, and in fact sort of the invention of the concept that you could do, you could breed natural variation together to produce a more useful crop. And then in the modern era, we've taken that same principle and through the application of sort of a more scientific method, created corn that has much higher yields than these initial efforts. Okay. So where we are in biomass today is we're sort of at the stage where we were 5,000 years ago, in that we've identified a few very rapidly growing kinds of plants that might be suitable as feedstocks, as materials that we could grow in large quantities and convert into fuels. But this took several thousand years to, I know I said 2,000 here and 5,000 there. This took several thousand years to occur. We don't have several thousand years in terms of the need to deal with this energy problem. So we'd like to go from identifying the potential candidates to jumping right away to this modern genome-enabled improvement so we can try to figure out how to do this efficiently. So there was a study that went on for several years, sponsored by the Department of Energy and the US Department of Agriculture, to identify candidate crops that could be grown as a fuel stock or a biomass feedstock. So these are plants that grow naturally in different parts of the country that have the potential to be generating a significant amount of biomass in a way that could impact significantly on our fuel consumption. And so they, not surprisingly, depend on which part of the country you're looking, but there are various kind of prairie grasses like switchgrass. You may remember that was something that President Bush mentioned in his State of the Union address a couple of years ago. Switchgrass shows up also here in the southeast and also in the Midwest. So switchgrass has a wide range here. Poplar trees are also very rapidly growing, and there are various experimental plantations of poplar trees. You can grow a poplar tree to sort of maturity in 5 or 6 years. So that's another example of a potential feedstock. And if you stare at this carefully, you'll see that there are some other kinds of grasses. Sorghum is very closely related to that very tall grass that I showed you before, as well as to maize. Eucalyptus shows up on the list. These are the ones that seem like they might have potential to be grown sustainably. Okay? Not that it's necessarily been done yet, but this is what are sort of like the Native Americans wandering around identifying, hey, this is a potential for something that I might be able to eat. These are potentials for things we could work with for energy. Okay. One thing I want to emphasize in this discussion of genomics is that genomics is not genetic engineering. They have the same root. I mean, gene is part of both of the names. Genomics is basically this integrated understanding of the biological system using the knowledge of the genome sequence. Genetic engineering is modifying that sequence. Okay, and I'm sure there'll be people who will have questions about this, but you can use genomics to accelerate traditional breeding processes in a way that I think I'll show you in a little bit. And the main thing is that we have to learn a lot more about these plants than we do. And so the genome sequence, in the same way that the human genome sequence is really accelerating biomedicine, the genome sequence of these plants is going to accelerate plant science, and it's going to have benefits not just for bioenergy, but also for agriculture and food production as well. Because we understand extremely little about how plants regulate their growth, how plants respond to various pathogens, and why it is that some plants are really hogs of nitrogen, they need a lot of fertilizer, whereas others are really much more efficient in how they take advantage of those nutrients. So this is a picture of the Genome Center. This is some of the staff of the Genome Center, and this is a poplar tree that we planted because we sequenced its genome. And so the idea is to have a little garden of plants whose genomes we know. As David mentioned, this is about 20 minutes away from where we are now, just driving through to Walnut Creek. And it has pretty good sized staff. It was responsible for generating about 1/8 or 1/9 of the human genome sequence as part of this international collaboration. And then when that project was completed, the other centers continued to do biomedical research, but since we were funded by the Department of Energy, we focused our attentions, our energies, on what we could do to use genomics to further bioenergy research. Okay. This is a facility that's managed by LBL at the J and the JGI, the Joint Genome Institute. It's joint because it involves both Livermore, Berkeley, and Los Alamos, and also other partners like Oak Ridge and Pacific Northwest Lab. So it's really kind of a central facility for all of the DOE and non-DOE users, and it's part of, and through this, it's part of the Helios project. Okay, so I now want to give you a few vignettes about how are we doing with this process. So the basic theme is we want to start with energy from the sun, make cellulose, convert that to sugar, and then break it down into alcohol. And so there are three basic biological components of that. One is we need to grow a lot of cellulose. That's this sort of feedstock idea of plants that are generating a lot of materials. Then we need to convert that plant material into something that is simpler, to break down the cellulose and lignocellulose into sugars. And then finally, we want to convert those sugars into alcohol, and that's done by yeasts. Okay. And in the middle case, I'll talk about some work that's going on at the JGI on looking at the termite, which is, after all, a very efficient degrader of plant material, as many of you might unfortunately know. Okay, so in the case of teosinte, once you realize that you can eat the little ear, we want that ear to become more robust. You want more of the plant's energies to be devoted into making the thing that you're going to eat. So that's a fairly straightforward measure of yield. That's the thing you want to optimize, whether you're doing it through sort of a traditional breeding method or something that's souped up with genomics. What are the things that we want to change about a bioenergy feedstock? What makes a bioenergy feedstock a better source of material? Well, Jerry Tuskin, who unfortunately couldn't be here, has spent a lot of time thinking about what it would take to domesticate poplar as a biofuel crop. And there's a bunch of things that you want to be able to influence. You'd like the plant to grow very rapidly. You'd like a lot of the material of the plant to be in the trunk, because it's easier to process this afterwards. You like the root system to be very compact, because that's material that's harder to access, and you might want to pull a whole tree out of the ground. So you want to have it be stable, but you don't want to store a lot of materials here. And you have various other things that you would like. You'd like it to be pest resistant, because you're going to want to grow these at very high densities. You like the density response to be robust. You like not to have to water these things if you can get by with it, so you'd like to have minimal water needs, and so on and so forth. So basically, you want to get something for, if not nothing, as little as possible. So we teamed up with this international group, a group from Canada, Sweden, Belgium, and other partners, and sequenced the genome of the poplar. And I could start reading the genome sequence to you, and it would be a string of A's, C's, T's, and G's, and it would take a long time and not be very informative. What I want to do is show you a little bit about how Jerry and his team are using that information to accelerate our understanding of poplar growth in particular. Okay, and there's lots of other stories here: the drought resistance, the root distribution, the way the plant is shaped. All of these things are things that people are studying right now. But I have a very short vignette about how it's working in terms of growth. Okay, so these are some of the goals that Jerry has set, but one of these goals here, that's not fair, okay, is how do plants control their growth? And so there are these plant genes, auxins, and auxin response factors. Okay? And these are genes that are basically growth hormones and growth hormone modulators for plants. Okay? In the same way that human growth hormone and all the stuff that Barry Bonds may or may not have taken is a modulator of growth in humans. Okay? So we'd like to understand what are these plants, these genes. And one of the things that you learn when you study genomes is that it's actually very common to find that a gene, there's not just a single, there is a single insulin gene, but there are also many insulin-like factors in the human genome. Okay, so the idea is that there are many relatives that maybe they have overlapping functions to them. And there's an insulin receptor, and there are other kinds of insulin-like receptors. Okay, so it's often not the case where you could say, well, I want to find the gene that's involved in plant growth. It turns out that there's a whole bunch of genes involved in plant growth. Many of them are related to each other. And you can recognize by looking at the sequence that they're part of a larger family. And so I could give you the list of these genes. And it turns out that poplar has 40 genes in this auxin response factor category. So what else do we know? Well, poplar is not the only genome that's been sequenced. A very small weed, it's about this tall, you can grow it in high numbers in greenhouses, that sequence is also known. That's not something that's going to approximate very well a rapidly growing tree, okay, which is woody versus a little herb. So if you compare the genomes, you find that both the model plant, Arabidopsis, and also the one we're looking at, poplar, they have many genes in this family. Poplar has 40, Arabidopsis has 23. The number is not so important, but the key is that we don't necessarily know what any one of those 40 do, but we know that they're involved in growth modulation. But now that we have 40 of them, and we know there are 40, there's no more than 40, there's no less than 40, we can go in and do a systematic series of experiments, 40 experiments, looking at what happens if you knock down the expression or remove that gene from action in poplar. Okay. And so Jerry and his colleagues are doing that. And here's I'm changing a little bit between the two gene families. Don't worry about the details. It all works out. But if I look at a poplar tree after a few months of growth, I get a certain radius. And if I change the expression of one of these genes, now I get a tree that's growing at a much more rapid rate. More rapid growth means more photosynthesis, means more carbon dioxide coming from the atmosphere, more plant material produced, more efficient biomass creation. And here's just the sort of cross sections. You go from a diameter of 4.4 centimeters to 7.1 centimeters. What's important here is not this particular case, but the idea that we have now a complete list of the genes in poplar. And so you can do these kinds of systematic experiments extremely rapidly. So that's one of the advantages of genomics, is that you can very rapidly scan for genes of interest. Now I can go, and this is one of the things that Jerry's in the process of doing. Now you can go out to a poplar forest, and you can say, I want to find those poplar trees that are going to be good for breeding into my plantation. I want to bring this feature of rapid growth into my biomass producing garden. Knowing that this gene IAA 16.3, whatever that may be, knowing that that gene is one that is relevant for the rapidity of plant growth at this point in development, I can go out and look for naturally occurring plants that have that variant in them. Okay, so I'm not doing any genetic engineering here. I'm just using what I know about how plants function from the genome to go out and find these naturally occurring variants in a way that's not that dissimilar from Native Americans trying to figure out which of the source teosynthase they should breed together to get something that is more effective for their purposes. Okay. Another aspect of this that I think is very important to the Helios program and to the JGI is to think about the plant not just as a unitary thing, but as also part of an environmental system. So when we sequence the poplar genome, you basically take plant material, you grind it up, you purify the DNA, and then you figure out its sequence. We found a whole bunch of other sequences in there too that were not from the plant. They were from various fungi, various bacteria that live in and around the plant. Okay? And these are not just errors in our sequencing process. These are the natural biota, the natural organisms that live in the vicinity of this plant. And in fact, it's been shown that if you change the fungi that live near the poplar trees, they don't do as well. Okay, so it's an integrated system. If we're going to do things sustainably, we need to figure out not just how the plant responds, but how all of the rest of this community, many of which are sort of anonymous at this point, are distributed. And one way to get at that is, since all organisms have DNA in them, one way to get at that is just to sequence. And so in the process of sequencing what we call the poplar metagenome, not just the genome of the tree itself, but of all of the other organisms that live in and around it, because they're part of the system too. Okay. One of the things that you learn when you think about plant biology and how plants are related to each other is that in the same way that humans and dogs are related to each other, plants, different kinds of flowering plants are related to each other. And that's actually kind of almost a numerically accurate sort of statement. And so here are some of the plants that we're sequencing, not necessarily because these themselves are going to be biomass candidates, but because by sequencing them and comparing them, we learn a lot about plant biology. And we have ways in which we can transfer the information between different systems. And so the soybean genome is one that we've sequenced, nominally because of its potential value as a biodiesel crop, but also, obviously, it's one of the major crops in the US. Mimulus is a very beautiful plant that grows in a very wide variety of different environments. It's known for, it can live near a toxic mine, it can live in high drought conditions, it can live underwater. Different variants of these plants live in different environments. If we can understand why that is and how they differ from each other genetically, we could use that information to help our understanding of plants in general and then feed that back to the feedstocks. And then Brachypodium here, this is a little tiny grass. You can see the size of it. You know where Target is by the freeway? There's an enormous building that says USDA on it. This is a collaboration with the USDA Western Regional Research Center trying to develop Brachypodium as a genetic model for grasses that might be useful for bioenergy. So I can go, there's a story involving all of these, but I just wanted to give you a few of those vignettes. This is a variant of that picture that I showed you. Again, this is a regular-sized person, and this is a stand of miscanthus. These are, one of the things that's interesting about this is that these are perennial grasses. So unlike corn, which you have to plant every year with seeds that you have to buy from the seed company, these are plants that you cut them off, and the roots can just grow back the next year. And because they have that perennial lifestyle, they're also particularly good at storing nutrients. So when you cut them off in the winter, all of the nutrients have been retracted to the roots, and they're there again for the next growth in the spring. Okay, this is a natural thing that the plants do, but it's particularly useful to us because it means we don't have to use nitrogen fertilizer. So all of the nitrogen, not all, but the nitrogen fertilizer effects that are so dominant in growing corn are likely to be much lower here. And the key issue is, what are the impacts of this in the long term? And so to answer that question, we're collaborating with Steve Long and others at University of Illinois to try to understand that. And they have plots of these grasses that are growing, and they're going to continue growing them for several years. We're going to be sequencing the genomes and the genomes of all the stuff in the soil to really try to understand what these long-term effects are. Because in the end, that's what we want to make sure that we're doing something right. Okay, I have a couple more vignettes. It should be maybe 10 more minutes. Okay, so now let's say we've figured out how to make feedstocks grow very rapidly with no inputs. Now we want to convert them into something that we can use as a fuel. So one possibility is just that you just burn them right on the site. You bring them to some local burning center where you incinerate them, you collect the particulates so you're not contributing to that kind of pollution. You restore the carbon dioxide back to the atmosphere, which is where the plant got it in the first place, and you now have heat energy that you can use to drive turbines or do whatever you want to do. That's not really going to help us so much in terms of transportation. So the advantage of ethanol and other kinds of liquid fuels is that you can transport them and put them in your car and drive around with them. Okay, so one of the big focuses is trying to figure out how transportation fuels can be basically retrieved from the plant material. And you don't have to look very far to find organisms that are really good at degrading plant materials, and termites are one of them. And so I'm going to need to, if you could advance it when I ask. So this is a collaboration that's been led by Phil Hugenholtz, scientists from Diversa Corporation, now Verenium, and Jared Ledbetter from Caltech. And so they were asking the question, how is it that termites are so good at degrading plant material? And it turns out that in a way, it's not the termite. The termite is just the home for this process. So I think I did that. That's good. So this is another view of what, at the molecular level, woody plant material might look like. It's that cellulose that I showed you before, plus another material called lignin and hemicellulose. And the termites, oops, I think it's got to go the other way. The termites ingest this wood. They grind it up with their jaws, and then the woody material ends up in their gut, where it's then broken down. And this breakdown occurs not from enzymes that the termite makes, but from organisms that live inside the gut of the termite and take advantage of the termite as a wood harvesting organism. So it's kind of like the bacteria that live inside the gut of a cow. Right? The cow is a very efficient delivery mechanism for taking grass from the pastures and delivering them to the bacteria that live inside their gut. We'd like to understand that process. And so Phil and Jared and their collaborators, in conjunction with the government of Costa Rica, went to gather sort of monstrous termites from the Costa Rican forests. And you can go to the next slide. So it turns out if you grab the front of a termite and the back of a termite and you yank, you will pull out its gut. Bet you didn't think you would learn that kind of useful information from this talk. And so here's a view of one of those guts. And if you stretch it out, you can see that there are several different chambers. And there's one chamber here, this P3 segment, which is known as where a lot of the enzymatic degradation of the wood goes on. Okay, so what Phil and the collaborators did was they did this with a couple, keep going, please, 200 of these termites, and used the syringe to extract the material that was within that gut. Now, in that gut is a bunch of woody material, but it's also a bunch of bacteria and other microbes that secrete enzymes that digest the material. Okay? So the microbes get a home within the termite, and they get carried around from tree to tree. And the termite gets to take advantage of that material that's been degraded by its commensals. Next slide, please. And so out of this came the discovery of over 200 different enzymes that are on the left here. These are the plant materials, cellulose, cellobiose, and so forth. And these are different kinds of enzymes. The names won't necessarily mean anything to you, but these are proteins that are made by the bacteria that are very efficient at breaking the bonds in these polymers and giving rise ultimately to sugars. And it's these sugars that then can be fermented. So the purpose of this is to break the plant back down. Next slide, please. So now we've got it broken down to sugars. I want to end with one very, very small vignette that maybe some of you could tell me exactly what it means. This is sort of at the frontiers of what we understand. There are many microbes that will, starting from a sugar solution, produce alcohols as part of their natural fermentative metabolism. And so the next slide shows one of them. This is a yeast that's actually related to Candida, some of which cause human disease. And this is a particular yeast that is able to degrade one of the sugars that I didn't show you on the last slide. It's a sugar that's specifically a part of biomass at a low level. And it's a 5-carbon sugar, for those of you who are chemistry aficionados. Okay. And so what happens is this, when you start with a mixture of this xylose and you add these yeasts, the yeast will happily take up the sugar and produce ethanol. Okay, that's a good thing. And eventually they produce so much ethanol that they die. Right? So what we'd like to do is take advantage of these guys and have them be more efficient at producing ethanol and more tolerant of ethanol. Okay. They're already doing it. We'd like to make them better at doing it. And so what Tom Jeffries, our collaborator at the University of Wisconsin and the Forest Products Laboratory there, did was he took one of these naturally fermenting yeasts, put it in a bioreactor, and then allowed it to evolve. Okay, so if you take a bunch of naturally occurring yeasts, some of them are going to be better at fermenting xylose than others. And under the right conditions, you can get it so that in your lab, you sort of mimic the process of evolution, where the ones that are better are the ones that survive. Okay, so we started with a reference strain of Pichia, and then evolved it forward over the course of several years so that it was 10% better at fermenting xylose. And 10% may not seem like a lot, but if you follow some of these discussions about is it worthwhile to make biofuels and what are the economics of it, it's going to be a lot of these sort of 10% changes that will make a difference. So 10% in this case is actually a lot, and we'd like to understand what makes this new Pichia better at fermenting xylose than the old Pichia that we started from. What happened during this process of evolution in Tom's lab? This is, again, not genetic engineering in any real sense. He basically looked at mutants and found the mutant that was the most effective at doing this. So the natural question is, what changed? What were the mutations that allowed this particular yeast to be better at fermenting xylose than the starting material? And so now we're able to take advantage of the fact that the human genome sequencing has gone on to the next level. So now that we have the reference sequence, what everybody really wants to know is, what does my genome look like? What does the genome of my cancer look like? Okay, these are the kinds of questions you'd like to be able to answer. You don't want to have to spend billions of dollars per genome. That's not very cost effective. You'd like to figure out how to do this for a couple $100 or at least a couple $1,000. And so there was an article in this weekend's New York Times about various companies, including many in the Bay Area, that are trying to figure out how do we really cheaply sequence human genomes. Okay. If you have a cheap way of resequencing a genome, knowing a reference, finding out what are the variations, you can apply that not just in the human case, but you can apply that in many different cases. So these are some of the new instruments that these numbers may not mean anything to you, but this is a third of a human genome per run at the cost of about $5,000. Okay. So these are new technologies that are really going to change. Certainly they're going to change biomedicine, but they're also going to change bioenergy. So the next slide. So this is where I need your help. These are the 15 differences between the original xylose fermenter and the extra good xylose fermenter. And I think if you click again, you'll see some things highlighted in red. This one we knew about. These other ones, we've stared at this for a long time. They're not obvious. It's not clear what is making this yeast better at doing this. But at least now we have a finite list. I mean, again, as I said with the poplar case, this is a very well-defined set of changes. And these are the only changes that have happened. So somewhere in here is the clue to making xylose fermentation better. And this is the kind of thing that genomics promises us. By having the complete enumeration of changes or genes or other features of an organism, it really kind of constrains our future analysis so that we can get much more rapidly to the heart of the problem and solve some of these vexing questions. So this is kind of an overall summary. The whole point of what we're trying to do is to figure out how we can use the natural energy from the sun and the carbon dioxide from the atmosphere to produce rapidly growing plant materials in a sustainable way that then we can break down and convert into a fuel that when we burn it is just going to be restoring carbon dioxide to the atmosphere where it came from in a way that where the whole thing is sustainable and economically feasible. And if we can figure out how to do this, then it becomes up to the policymakers to figure out how to implement all of this in a larger context. What we can do is we can provide the technical know-how to figure out if and how it can be done. So I think that's all I have. Thanks for your attention. I probably have two more slides. Okay, I don't have any. Thank you.
questions, and I would encourage you to seek out the microphones that we have in the quarters of the audience here, and if you could just limit your questions to one at a time and allow other people to chime right in.
Do you want to be the MC, David, and
Sure.
pick people out? We have one over here. Why don't you start?
Are there other things in the ground that have to be used to make the plant grow besides carbon and sunlight?
So in order to be able, yeah, so the question is, are there other things that are needed besides carbon and sunlight? And I tried to mention those. So certainly the kinds of nitrogen inputs that come from fertilizer now, phosphorus, potassium, there's a lot of other elements that need to be contributed in order to make a viable plant. You have to, the whole issue of sustainability is how do you deal not just with the easy part, which is the carbon cycle, but all of these other nutrient cycles.
I don't know if this works, but the question is, you mentioned,
It is working, I think.
it is already. You mentioned that eucalyptus was a good source of biomass, and up in the hills here we have a lot. The university is getting in a lot of trouble again because they're talking about cutting down most of the trees and probably messing up the whole environment while they do it. Are you planning on using some of those eucalyptus trees in your studies, or are they just...
What we're doing with eucalyptus is a collaboration actually with a group in South Africa and Australia, and it's not related to any of the local land use issues.
Well, would you talk to them so they don't just waste things and denude the hillside? Thank you. All right.
Traveling up and down the coast, the Pacific Northwest, I see this grass that has these yellow flowers. They are called the yellow flowers. They grow like crazy. My question really, it has something to do with what is the difference between genomic manipulation and genetic engineering?
So the question is the difference between genetic engineering and genomics. So when I define genomics, it doesn't involve any manipulation. It involves really understanding the basic genetic information that's inside every organism. And with that understanding, you can do a lot of experiments that don't involve any sort of manipulation at all, that really help you understand the biology. Now, there's a separate issue, which is being able to modify that genetic code. And that's really, I think, a very separate issue from the one that I was talking about today. I'm sorry, other questions?
Wait a second, I was talking about the yellow grass I was telling you about.
Yeah.
And I worry about, because it seems to be very invasive. Mm-hmm. They grow like crazy, and people are having an extremely difficult time trying to eradicate them, mm-hmm. because they have become, a pest, literally. Right. So.
There's an important issue there, which is this issue of if you want something that grows very rapidly and grows without a lot of inputs, then you're sort of defining a weed. And so you have to make sure that what you're doing is not going to displace other plants. And that's, I think, part of understanding the life cycle of these plants and how they function.
Just wondering, what's the highest photosynthetic conversion efficiency that you've gotten? Because when I looked at this maybe 10, 15 years ago for macroscopic plants, not algae, but what we're talking about today, it was about 2%, which is kind of puts a limit on the whole efficiency of the entire sunlight to liquid fuel process. Are you doing much better than that now?
Elaine may have a better sense of that.
Comment. The actual efficiency that we're interested in is the efficiency of the crop, not just the process of converting sunlight into, 1 photon of sunlight into one equivalent amount of energy, 1 unit of energy. And with a crop, you have to average over the whole year, and things of that sort. And most plants get about 0.025% efficiency. This miscanthus is very close to 1% efficient over the year. It's pretty remarkable. There aren't too many plants like that. So that's why it's a very popular, subject of.
I think the other point is that sounds like, well, it's 99% inefficient. How could that be? How could that be good? And I think the point, the counter to that is there's a lot of energy coming from the sun. And if you can capture 1% of it, this report that I referred to where I showed the map, even at the kinds of efficiencies that you get with just naturally occurring plants, if you could efficiently convert that into transportation fuel, that would be enough to power a significant fraction of our transportation needs. So you don't need to be 100% efficient. You just need to have something that's sustainable.
I have a question here.
Where are you?
I think I remember in the beginning you were talking about limitations in using ethanol.
Yeah.
About transportation difficulties or things like that. So my question is, why, considering all those limitations or difficulties or whatever, you are still working apparently to produce ethanol?
So the question is, why ethanol, given that it has various imperfect features? I think it's a good question. The one good feature that ethanol has, and also biodiesel from soybean or other oil sources has, is that it can be used with I guess you call it the fleet, with cars and trucks that we have out there on the road right now. So let's say that we figured out some next generation fuel that our current cars could not run on. Well, we'd have to wait until those cars were retired, basically, and people would have to, in a systematic way, purchase the cars that could run on the new fuel. So one of the advantages of ethanol is that you can mix it with gasoline at some rate. 10% is pretty common. And in that way, take advantage of the existing fleet. So it's kind of a transitional fuel in that sense. People have looked at butanol as a higher density alternative to ethanol, another short alcohol. And then looking at the longer chain oils, such as some algae produce, may be possibilities for the future. So there's a lot of effort. I mean, ethanol is something that's sort of kind of potentially accessible on the near-term horizon, and certainly we want to be able to do that efficiently, not necessarily with corn kernels, as I described. But these other potential future fuels, they're also part of the Helios project and the other bioenergy projects that are going on.
So you mentioned that there are these other organisms that are mixed in with poplar, and the way you sequence the DNA or discover whatever, you grind it up and purify it. How do you tell which DNA belongs to the poplar and which belongs to the other organisms?
So, my friend Simon and my group, I told them not to come to the talk. It's these guys that do the work. It's we can take advantage of the fact that we now know the genomes of several thousand different organisms from various work that we've done and other sequencing centers have done. So if we know the genome of a bunch of fungi and we sequence a little snippet of a genome that is embedded within poplar, if we compare it out there to all the other sequences, we'll say, hey, this is much more similar to a known fungal sequence than it is to a known plant sequence. And in that way, by the similarities of the sequences of the letters, we can have a pretty good idea that it's not part of the plant, but it's part of a fungus that lives near the plant, or a bacteria. So it's by this comparative method that we try to understand where these little pieces come from. Then you can go back and with a hypothesis, oh, this should be part of the fungal complement that lives on the leaf or lives in the litter around the tree. Then you can go back in and do a hypothesis-based test of that by trying to isolate the organism that had that. And that's been successful not in the poplar case, but in other cases, we've been able to anonymously sequence organisms that live, for example, in very toxic environments that had never been cultured, and then go back. This is work with Jill Banfield and her group at the university. She's been able to then go back, knowing the genome, she can then figure out conditions under which she can cultivate these organisms. So a lot of organisms out there that nobody knows how to cultivate.
Question over here, Dan.
In harvesting the sun's energy through photosynthesis, is that essentially an extra step from harvesting the sun's energy directly through solar, which I guess would be through physics?
If we had sort of a purely electrical way of capturing that kind of efficiency, then that would be an alternative, right? You could put out solar panels and store, if we had good batteries also, then you could store that energy in batteries and drive it to a, probably still would call it a gas station for a while, but you pick up your battery and exchange the old one. The solar technology and the battery technology are things that are a big part of the Helios project. I think if it's all going to work, we have to take advantage of all the different kinds of energy sources that we can get. So, and of course, conservation is going to be an important thing. So it's not going to be, this is not going to be the only solution. There's going to be conservation, there's going to be wave and geothermal energy, there's going to be a lot of solar, but all of this needs sort of scientific and technical development, and that's the essence of the Helios project.
Hi. In your pursuit of easy cellulose from plants, have you ever considered that perhaps the scientists will not be able to convert cellulose to sugars as they had hoped?
So the question is what if we just can't do it?
What if you can't do it? I understand it's 7 to 10 years.
So I think we have to try. I think what we know from understanding of this kind of biology and others, there may be technical limits, but we certainly haven't reached them. An example I gave of the yeast that ferments this xylose sugar, even that yeast had an extra 10% activity left in it. And so if you kind of work out the numbers, what do you need to get to make this be an economically feasible process? We're not that far off. We need, maybe a dozen of those kinds of advances. So I think we're close enough that I'm optimistic, but it's possible there'll be some fundamental limit. We have to try, and we have to try on these other fronts as well to figure out how to deal with this problem.
Producing alcohol from sugar fermentation process, some carbon dioxide is released during that process. Is that significant in making greenhouse gas?
I think so the question is, what about other carbon dioxide that's released during this process? And I think the main point is that if you do the accounting right, you have incorporated carbon from carbon dioxide into the plant, and then you may liberate some of that carbon dioxide in the processing step. That's fuel energy that's an inefficiency of the process. But I think in terms of the net balance of carbon dioxide in from the atmosphere and then out during processing and during the driving around in your car, I think the net balance has to be the same. So I don't think that has a negative effect. Yeah. I mean, what is significant is the carbon that goes into producing the fertilizer and transporting things around, and all of those kinds of accountings, people are still working that out.
Yes, any of the processes that you described are all going to have residuals, waste products left at the end. Have you given any thought, or would you discuss what you're thinking about and doing with them?
Yeah, so when you, I mentioned that the woody materials have lignin in them. And so at the end of the day, you're left with ethanol, but you're also, as you say, you're left with the ash and other things. So the question is, can you productively use that, those byproducts? Or you have to figure that into your accounting. And I haven't thought much about that. Jerry Tuskin, who's the plant biologist on the project, he's thought quite a bit about it, and that you can use that material as part of the fuel for the process of conversion in a way that makes sense. But I don't have the answer at my fingertips.
Any comments about the recent papers in Science that indicate that carbon emissions may increase due to land use issues?
Yeah, I think that's a really important. Those kinds of studies are part of this larger, Helios portfolio. Certainly, the Energy Biosciences Institute. I was at a meeting of that a couple weeks ago, and there's a lot of interest in making sure that it comes back to this issue of sustainability. So what that science paper argued is that if you are going to get cheap energy from planting these kinds of crops, you might have the unintended consequence of clearing out forests and other sources, other sinks of carbon in your rush to get more land for this purpose. That's all part of both the sustainability issue, but also the policy issue. I mean, if the policies allow that to happen and there's an economic driver, then it's going to happen. So if we recognize that it's a problem, I think that's has got to be in the policy arena, whether you're doing it through some kind of carbon cap or carbon tax, or however you choose to do that. You want to provide economic incentives for people to do it in a way where the net effect is, as it could be, positive.
Hello. My question concerns algal oil, using algae for oil. And you have more research access than I do, just using Google. It appears that the Department of Energy spent quite a bit of time and money, say maybe about 10 years back, researching this, but then it just sort of stopped. And that most of the current research has to do with selling algae pills to people as a, rather than as a fuel. Would you comment on that, please?
So there was, as you say, an investment of the DOE in investigating algae as potential sources of biofuels. Many algae produce oils of various kinds that if you could figure out how to do this in bulk, you could potentially produce oil-based fuels. That early study, the one that you referred to that ended about 10 years ago, I think it was basically because the money ran out. It was not viewed as being the most productive way to go forward, and the cellulosic sort of continued. Now that there's more, with the cost of oil rising and the more attention paid to this kind of problem, there are at least a dozen biotech startup companies working on algae, and we're discussing projects that we can do with them to sort of facilitate their genomic-enabled study of these things. So people are going back to this and looking at some of these algae as potential sources of fuel. There's a bunch of issues to address there. One of them is if you just grow algae in open vats or in churned up vats, they tend to be, if you find one that's really good for biofuels, it tends to get contaminated with other stuff that gets in there. So there's a kind of a pathogen resistance issue, if you want. So there are a lot of technical issues there too, and we're working on those. I just didn't have time really to talk about it, but I think it's a good direction. I'm excited about it.
We'll take a couple more questions, and then I'm sure Dan will stick around for a few minutes to field your questions personally.
Sure. Just a quick one on the amount of water that's consumed in the entire process from, I suppose, planting all the way to the production of a gallon of biofuel. And I've heard figures like it requires 6 gallons of water to produce 1 gallon of biofuel. Is there any way to diminish that? That just seems like a fairly heavy price to pay in terms of embedded water.
I think you're right that water use is going to be one of the big limiting factors, especially if we try to do this on a big scale. And I really don't know. Somebody talked about what are the potential limitations. I don't know if there's a sort of. I don't know how close we are to sort of the practical limit on that, or how much we can reduce that use. That's a good point.
Isn't this discussion totally ignoring the political aspects and the big business and people driving cars that are much bigger than they need to? Wouldn't a lot of these problems be solved with perhaps a higher tax on fuels, with maybe rebates for the
So I totally agree that conservation is going to be important, and also making sensible use of the resources that we have. That's not something that we're going to be able to necessarily carry over from anything that you might personally, or we might personally in this room want to do, to large growing economies like India and China that you can't necessarily legislate that they're going to use less oil than you had a chance to use when you were a kid. So I don't know how that's all going to play out. It's clear that there's going to be increasing demand for energy, and we want to be at the forefront of both the conservation aspect, but also figuring out new ways of getting that energy into not just our hands, but everybody's hands globally. That's a good point. We certainly should not overlook that, but I don't think that just being conservative in your use of energy is going to really make the kind of impact that we need to make over the long term.
One last question, and then I invite you to stick around for a little while, and Dan will try to answer your questions.
I was wondering, like, compared to corn, how efficient is sugarcane in ethanol?
Yeah, so sugarcane is a close relative of this rapidly growing plant, and it's a very efficient source of sugar directly, or as opposed to having to produce it from starch or sugar or more easily broken down starch. I don't have a number off my fingertips of the relative efficiency of sugarcane over corn, and it involves discussions of subsidies and other sorts of issues. But sugarcane is a very attractive non-cellulose-based biofuel source. But in Brazil. So I think we have to, it's part of a larger discussion.
Will you please join me now in thanking
Thank you.
The next stop on the series is Monday, March 10th, and the speaker will be Marianne Kietz on saving power